More important ideas about variable expressivity is the phenotype is always present. It's just a variable phenotype. We can't use the word variable to do some other word other than variable. Here I chose different instead of the word variable. You can't use the word in its own definition. It's the basic rule that I'll always go by. So variable expressivity, the phenotype is always there, but there are degrees to which the phenotype is exhibited. An example that was used in class is polydactylism. You can have more than 6 fingers, be on one hand or the other hand or both hands, or one foot, the other foot, or both feet, or all things could have extra digits. That's the variableness of the expression. You always have extra digits. It's a question of how many extra digits and where those digits are actually located. For penetrance, we do see a true unlinking. I don't use the word unlinking, but it is a sort of an okay word to think about. The phenotype and the genotype are not closely together because you can have a genotype but not have the phenotype. So because of this idea of you have the right genetic background for it, but you still haven't exhibited the phenotype, is the idea of penetrance. And penetrance is a probability, which means it needs to either be a percentage or a 0 point something number. And I will be very strict on the exam. If you write 49.53, or whatever the answer was, don't put percent on the end of it, I'm going to count it wrong. Because the penetrance can never be more than 1. Because it is a percentage, we do want to keep in mind that 100%, the percent is important, 100 and 1 are really different things. So just please keep that in mind as we go forward. A lot of things in the calculations we're going to do are actually percents. So it's good to get in the habit of writing percent on things or leaving it as the decimal point number that you actually calculate. It wasn't many people, but it was at least 3 people, so I made a note. I didn't say, please help me out, follow the directions, but I did say, please follow the format that I posted in the question. Other than these 2 questions, does anybody have any questions about anything from the quiz? Yes.
The epistasis question, I don't remember exactly which one it was, but that was one.
There was 2, and they ended up being the exact same.
Yes, that question.
And there were 2 questions about introns, or sorry, exons, that were the same, they're supposed to be different. I'm still struggling with rewriting my quizzes, apparently. But those questions should have been regraded and should be graded now correctly. Those questions were about the 9-to-4 to 3 ratio, in which case we have a recessive controlling a dominant. And so both those should be the exact same answer, and you should get credit if you have both answers. If not, let me know, I'll double check it. But it should say, one should say re-rated, the other one should say not re-rated because it was what it was normally. Same thing with the exon questions. One should have said not protein coding, and the other one should have said protein coding. It's a sad state on my mental faculties these days. I can change the question title and change the answer, but I can't change the actual question. I missed 1 of 3. I'm 67%. If I was in baseball, I'd be all-star. 2 out of 3. That ain't bad. There's a movie quote there. I can't remember what it is now. But it was the original Batman. I would be 2 out of 3 ain't bad. It's Jack Nicholson saying that. Any other questions about the quiz? Just one more quiz for exam 3. Sorry, exam 1. One more quiz before exam this weekend. Yes?
So like for the question where we calculated the penetrance and like you wanted it to like the thousandths decimal place.
I like 3 digits so I can see if you're doing it correctly, because sometimes you could get the right 2 digits and the wrong 3rd digit and have done the wrong calculation but end up near the right answer. So I just generally always say something like that on a question where it's 0.000. So you have 3 digits or 3 non-zero numbers, is what I generally try to say.
But like, did you want it left as like a decimal or to a percent?
It can be written either way. But if you write it as a percentage, it should have the percent sign.
Well, that was my, because like Canvas wouldn't let you submit it if you put a percent sign.
Okay. I didn't know Canvas didn't do that. The old version did. So I would just default leave it as a decimal point number and not worry about it. Any other questions? It's a lively class today. Everybody's ready for the cold weather and rain tomorrow? Yeah, it's supposed to. Hurricane. Yes. Hurricane in the Gulf Coast is giving us rain tomorrow. And on Friday, and on Saturday, and on Sunday. I think on Monday it's gone. But I don't know if you noticed, I need some rain. And some cold weather. I'm just looking forward to it. I love cold weather. Fall is the best time of the year. Summer sucks. Spring's okay. Winter, yeah, I like winter, fine. The fall is always the best. There's no debate. Don't try. I don't think I will have to speed up more than I usually talk to finish all the information we need. Before exam one, I think it's on Wednesday. Was it on Wednesday? I don't remember. Wednesday or Friday? I think it's Wednesday. I think it's a Wednesday.
It is a Wednesday.
If I did, I'm pretty sure I mentioned this before, but probably not. You never know. With me, I'm a little scatterbrained sometimes. And I go off on weird tangents, which is always fun. It's always good to get the professor talking about something that's what matters, right? Because then it's like, hey, no lecture day. But as far as I'm not trying to plan things out, I always try to leave the class period before the exam as the day to ask questions. Sometimes I may need to use some of that time for something, but for the most part, you can basically assume that the day before the exam is a question and answer day. So I'm going to talk about stuff, but I'll be happy to answer questions or go over things that you may want to go over. But if nobody's going to ask any questions, I will lecture for at least some of the class period. And I'll try to remind you of this as we go get closer to it, but it was on my mind right now, so I decided to share it with you. This is not a slide that's in your set of slides, although this picture is in your set of slides. I just decided to take away all the text and make the picture bigger because we're going to start to talk about telomeres and the unique telomere structure. We've already talked about the structure of the centromeres a little bit and repeat sequences. The purpose of the centromere is to facilitate the movement of DNA during cell division. For telomeres, their main function is protection. Protect the chromosome from loss of genetic information. It's not important to memorize the size of the repeats, that's just there for context. The telomeres are pretty long, but they're made up of a really small unit. And generally speaking, when we talk about telomeres, we are on the size of 5,000 to 10,000 in length. So how many sixes does it take to make 10,000? Don't worry about the math, but it's a lot, right? It's a lot. These are pretty short little things you have to keep making over and over again to build up on the end of your piece of DNA. It's not like a big block of something. It's a bunch of small little pieces of the DNA that are lined up at the end. When we talk about how DNA is replicated, we're going to revisit this idea of telomeres. And the basic idea is linear DNA, what do you do on the ends? It's hard to replicate when you get to the end of a linear piece of DNA. And so telomeres kind of help deal with that aspect a little bit. And so you really want to protect the euchromatin DNA. That's where all of our genes come from, so we use this heterochromatin repeat DNA on the end. Now, also with a linear chromosome, there is another potential problem to think about. So when we look at DNA, we typically just think about DNA as this basic piece of matter. It turns out there is a lot more complexity going on, both in DNA and at the end of DNA. This is a sad state for DNA molecules to look like this. And we'll get into why that is in just a second. But the telomere is not just these things, it's how the DNA is organized. And what I want you to think about, shown here on this slide, is that the telomere structure is going to organize itself into a loop. It doesn't matter how the loop exists, just that we basically have something that kind of looks like a circle at the end of the chromosome. And just for fun, we'll draw it like this. And it's not really important to know what it is. But the loop structure of the telomere makes the chromosome look like a circular piece of DNA. And in a circular piece of DNA, there is no free end. How many of you have seen Lady and the Tramp? What do the two dogs do? They grab onto the same spaghetti noodle and eat their way into a kiss. That's only possible because the spaghetti noodle is free on both ends. So without the telomere, the DNA is said to be free on the end. But if they're circular, or at least when there's this loop formed by the telomere structure, it is the protective thing that protects the ends of the DNA molecule. Now, what we protect from is digestion or degradation. And so without the telomere structure, the DNA ends will do something called breathing. It's a lot more molecular biology than we really care about. I'm not going to ask you to explain this phenomenon at all. But there is an important aspect of what happens when DNA doesn't have any structure at the end. Sort of hydrogen bonds go away for a little bit, and you do this thing where you kind of open and close naturally. When you open, you become single-stranded. And when our cells see single-stranded DNA, their first thought, virus. Kill this thing before it kills me. And the response to try to do that is to eat the DNA through using enzymes we call nucleases. They grab onto the end and Pac-Man their way down and just chew and chew and chew until they eat all the DNA that's single-stranded. So if you breathe, you lose some DNA. Go back together. You breathe again, you lose more DNA. Keep doing this, eventually you have no DNA. So the telomere makes this structure where everything's sort of folded and tucked in, so nothing's ever free, therefore DNA never breathes. But single-stranded DNA is typically a viral indicator, which could mean protect ourselves from being co-opted to make more viruses. So when the DNA breathes, the DNA gets digested. And then the DNA just gets shorter and shorter. So that's how we could lose genetic information. Without the telomere, you lose a little bit of DNA, then you lose a little bit more, a little bit more, a little bit more, eventually your chromosome is gone. And now you're a dead cell because you've lost an important piece of DNA. So telomere structure protects from a normal function. We want to protect ourselves from bad DNA, but sometimes our DNA that's good looks bad because of just genetics, not genetics, it's the thermodynamics and molecular mechanisms of how things work. But we're going to revisit this idea when we get to the idea of fixing DNA. What happens if DNA breaks? We need to be able to glue back together. And unfortunately, as we're going to see, breaking DNA is kind of like having no telomeres at the end. It's a bad state for the DNA to be in. If you don't get anything about this right now, I hope that you get more later when we talk about the repair process. This is a preview of why the repair process is needed and how it works as we go forward. Telomeres prevent the DNA from being digested, so we don't lose any DNA. That's really all we need to know about telomeres right now. Now, having said that, there's a little more to the story, and one of the fun things about the telomeres is they're made by an enzyme called telomerase. Isn't that nice? It's always nice when the enzyme is the name of the structure, rather than some weird thing like Sonic Hedgehog or Supersonic Hedgehog. Those are actual gene names. There is a disease caused by the gene Supersonic Hedgehog. I'm being literal here. And there's, as Dr. Smart, he knows the story, there was this mom somewhere that was really pissed off that no one believed her when she said that my child has Supersonic Hedgehog disease. That was a joke. Somebody had this funny idea, I'm gonna name this over Supersonic Hedgehog. Because Sonic Hedgehog has already been taken. And Hedgehog had been taken already. So it's a big long thing with jokes. Anyways, the enzyme is going to use an RNA molecule to make DNA, which is a fun thing to think about. That's reverse transcription. Using DNA to make RNA, transcription. Using RNA to make DNA, that's the opposite effect. But the cool thing about the telomere, or the enzyme telomerase, the RNA that it uses is always in the enzyme. Like it walks around with this copy of RNA that just copies it and makes DNA, and then slaps it on there at the end every time. So we call it an RNA-dependent DNA polymerase. It's always a fun thing to think about. But like a lot of things, this semester we're going to talk about enzymes that do certain things. This is an enzyme that makes DNA. That's really all we care about. The telomerase is an enzyme that makes DNA right now. One of the most important things about how's not the best word. The degree to which the telomere works is based on the length of the telomere itself. And I like to think about the telomere as like a biological clock. The telomeres will naturally start at a certain length. And over time, that length will get shorter and shorter. And at a certain point, the telomeres get short enough that the loss of genetic information becomes more likely to occur, triggering the cell to kill itself. We call this apoptosis. And so every cell has a ticking clock based off of how long your telomeres are. And every time you copy DNA, the telomeres get a little shorter. Or as your cells get older, the telomeres get shorter naturally. And what they typically think is a telomere length of about 10,000 or 10 kb allows the cell to divide about 70 times before those telomeres get too short to actually be useful. Now, when we think about telomere length, there is this direct line of thinking that scientists are trying to show that telomere length is directly correlated with aging. And if we can keep our telomeres for longer periods of time, maybe we can prevent some of the aging that we see in humans. There are companies that sell telomerase enzymes to try to call them anti-aging drugs. They're not exactly proven to be working, but what we do. But telomere length is an important signal for can you divide. So cell division and telomere length are going to be linked together. As telomeres get shorter, cell division becomes less likely. As they're long enough, cell division is very likely to occur. I don't want to put a number on it, at least right now. We'll talk more about cell division going forward. So we will revisit this idea of telomere length. This slide is a little out of place because we haven't talked about histone proteins yet. And this slide introduces a histone protein called H4. But generally speaking, again, when your telomeres get too short, bad things start to happen. And so it's not just telomeres that we have to worry about. When the DNA breaks in the middle, broken DNA is always a bad thing for the cell. It always is going to stop cell division until that thing is fixed. But when the telomeres get too short, there is a change in the protein content of DNA. And one of the histone proteins called H4 is going to be chemically modified through a process called methylation. So as your telomeres get shorter and shorter, the H4 protein that is naturally on DNA is going to be modified. So it's not necessarily the shortening of the DNA that's the signal, it's the modification of the histone protein. That's the signal that cell division should stop. But those two things are linked together. Shortened telomeres lead to methylation of something called H4, which is a histone protein, which allows DNA to be organized in a form that we call chromatin. This we can ignore right now. We're going to pick this up again with cell division. And there's a nice tie-in between the normal structure of a telomere and how it functions to other things that the cell has to do. But H4 methylation and telomere length and how they're correlated to does the cell divide or does the cell not divide is an important story that you should hopefully understand. And some of the things we've learned through cell biology and other things is that our cells start out with a lot of telomerase activity, especially early on. You need to crank out a lot of cells. And we start as one cell. That one cell has to be able to turn into billions of cells. You gotta undergo a lot of cell divisions to do that. So as we age, our telomerase activity decreases naturally over time. A lot of studies on cancers have found that in certain types of cancer cells, telomerase activity is unnaturally high compared to normal cells. And one of the defining characteristics of cancer cells is uncontrolled or unchecked cell division. They're dividing much quicker than normal cells do. So one idea is that one thing that could cause a normal cell to become a cancerous cell or tumor cell is because that's a mutation that alters the amount of telomerase activity that it has, making it divide past its point of no return. And so telomeres are too short, we shouldn't divide more, but hey, let's keep going because we've got extra telomerase activity. And one of the real problems with cancer is it's not one thing that causes cancer. Unfortunately, there are probably more than 1,000 different things, probably maybe 1 million different things that can cause cancer to show up. So how do you treat a disease when there's no one single cause for it? Makes it kind of hard. You know, I know it's gonna sound dark, sorry. I like dark humor sometimes. You know what the true cure of cancer would be? A short life. The longer people are living, the more cancer shows up. So as we live longer, I said this before, as we age, things break down and become less good, functionally speaking. And so cancer wasn't a big deal when people didn't live very long, but as people start living longer, we give ourselves the ability for these mistakes to sort of pile up. So you ever seen the movie Passengers with Chris Pratt and Jennifer Lawrence? The whole premise of that movie is one thing breaks and nobody's there to fix it, so that leads to another thing breaking and another thing breaking. Eventually the whole ship's gonna be destroyed. That's kind of like I think about cancer is one thing breaks. One thing by itself is not really bad, but it does lead to this other thing going wrong, this other thing going wrong, this other thing going wrong. And so maybe there is one trigger, we just need to find out what that one trigger is and stop it from happening, and we can do things. This is not really the one I want to show you. I Google's image of an actual bottle that said Telomerase anti-aging drug, not FDA approved, which I always like to do this. But this is a company where they're trying to sell you a plant-based compound which can help maintain and rebuild your telomeres. They say that they diminish as people get older. And the length of a person's telomere is a good indicator of his or her overall health status. Short telomeres have been associated with cellular aging and dysfunction. And the real biological age of a person's body may be more or less than their chronological age. This is something we'll talk about also later. So you may have seen somebody that looks way older than they actually are, or way younger than they actually are. And so your biological age and your chronological age always match. And so if you can figure out what your actual chronological age is, or your biological age, maybe we could predict when you would die. I don't know, we need to let them know when they would die. Might be helpful. Plan out your life pretty well, do everything you need to do before a certain date. Now, I brought up this idea early on this semester, but again, there were these two twins, and they were a good test group for the effects of prolonged exposure to space and weightlessness. So they sequenced the DNA of one that stayed on Earth, sequenced the DNA or analyzed the DNA of the one in space, and what they found was some pretty interesting things. And one of the things they found was telomeres were different. The changes were not permanent, which is also kind of interesting. That the changes that did occur went back to normal after a certain period of time. And so what they say is that because of space travel, the astronauts' telomeres got longer. Why would weightlessness or space make your telomeres longer? Don't know. Some people think it might be exposure to radiation because there's no atmosphere to protect you. Who knows? But there were some effects of being in space that we sort of maybe need to deal with if we're going to try to send people to other planets. Can't just freeze everybody and say, wake up when you get there, because that doesn't work right now. Cool if it did. What's that?
It'd be like Interstellar.
Or any other movie that freezes people, like Aliens, The Martian, yeah, they froze people there. There's some other good movies about freezing things. I mean, Star Wars, don't call it carbonite freezing, but still. Anyways, back on topic. Remember, chromosomes are not just DNA, but they're also proteins. One important thing to think about is the DNA that we find in a prokaryotic organism is actually too big to naturally fit inside the cell. It's too long to actually fit just by itself. The same thing is true for the nucleus of a eukaryotic cell. The DNA is actually too long and too big to fit inside the small container that is the nucleus. So the solve of this problem is to wrap DNA around proteins and make it take up much less space. We call this a compaction problem, or not problem, a process, which leads to where the word chromatin actually came into being, a DNA molecule wrapped around proteins in a specific formation. The compacted DNA is what allows it to fit inside the nucleus and to fit inside the bacterial cell and not take up too much space. But both bacteria and eukaryotic organisms will compact their DNA with proteins, but bacteria don't have histone proteins in the exact sense. They have things that are like histone proteins, but not exactly the same thing. The textbook that I don't require to buy has a nice little fun little analogy. Does anybody know how big a spaghetti noodle is? I've used spaghetti already with Lady Tramp. Imagine a spaghetti noodle. And that spaghetti noodle represents a chromosome that is 130 million base pairs long. If it were the size of a spaghetti noodle, it would not fit inside the nucleus. This is not the slide I was really thinking about. This slide. It's later on. It's definitely later on. The spaghetti noodle thing. We'll come back to it. To make the DNA fit into the nucleus, for example, we are going to reduce the size, but that increases the length. So if you squish something, it gets shorter, but it gets bigger or wider. And these numbers aren't really important, again, they're in gray. But still, the DNA has to be compacted in a way. Compaction actually makes it visible during cell division and visible during microscopy. We call the basic unit of chromatin the nucleosome. The nucleosome is DNA wrapped around a bunch of proteins. It's wrapped around twice, but that's not something that's as important as the proteins that are present. Every piece of DNA in your cells is already bound by a protein. This is an important consideration for later. I might talk about this in cell biology also. But DNA polymerase, the thing that's needed to copy DNA, or even RNA polymerase, the thing that's needed to make RNA, needs to bind to DNA to do what it needs to do. If there's already proteins in the way, important cellular functions can occur. So chromatin does have a benefit, making the DNA smaller to fit into the right space. It does make it harder for other things to use that DNA. So we do have to do a little bit of weird tinkering to get the things to work out. And we call the proteins that are naturally present on DNA in eukaryotic organisms the histone proteins. There's several different varieties of histone proteins to look at. There's 5 of them. So we don't have an H5, we have 2 H2s, just for fun. H1, H2A, H2B, H3, and H4. We already know something about H4, hopefully you haven't already forgotten. H4 is methylated, that tells us our telomeres are too short. H4 and H3 are proteins that are the most homologous between different eukaryotic species. Basically, they're the same. Every eukaryotic organism, H3 and H4 are basically the same, from bears to cows to insects to whatever. That's what I mean by most similar. There's hardly any variation from one species to the next species. This is probably true because of what H3 and H4 are used for. Every cell methylates H4 as a signal that telomeres are too short, trying to tell the cell it's time to stop dividing. H3 will do something important, but we'll deal with that in a second. So it leaves H1, H2A, and H2B to be really different. And H1 is the one that's the most different. And there's a reason for this one being so different in just a second. Histone proteins are pretty short, relatively speaking. Typically with proteins, we think about them being between 300 or 500 amino acids. So they're not significantly shorter, but they are on the shorter side. One interesting feature about the histone proteins is the abundance of two amino acids, lysine and arginine, which you can see are positively charged. So they want to hypothesize why it would be beneficial for these proteins to have a big positive charge component. DNA is negatively charged. And so if you're going to put or try to squeeze a lot of negative charges together, you better be able to get rid of those negative charges with positive charges. And so it's not just that we wrap the DNA around the protein, it's that the protein gets rid of the charge so things cannot be sterically hindered. You can have a guarantee that this question about the amino acids and the charges and the DNA charge are going to be on exam 1. It might be on quiz 3, but who knows? It's always on exam 1. It's a multiple choice question. Just give you all the different variety of things that happen. Just FYI. It'll be fun. Here's the answer right here. We'll skip that because you can look at it later and just answer it. Within the nucleosome, there are two parts. The core part, which is almost all of histone proteins, except H1, and there's two of each. The two of each part is not important. The fact that H1 is not part of the core is something I will ask you to remember. So here's the basic organization of the histone proteins in the ball that is the histone. And so this blue part is the DNA, you can see wrapped around it. How much DNA that's wrapped, not important to memorize. The fact that it's wrapped twice, not important to memorize. Slightly important to memorize that there's a part that's not wrapped by the histone proteins. This is the part where H1 becomes important. H1 will bind to this DNA that is between the two cores of nucleosome 1 and nucleosome 2. H2A, H2B, H3, H4 all have to bind together, which means you need to conserve what they look like so they naturally lock together correctly. H1 has no interaction with the others, so who cares if it's different? All it needs to do is bind to DNA. It doesn't have to bind to proteins or bind to anything else, just DNA. So the fact that these all go together is why we need to sort of maintain them to be similar to each other. So we wrap the DNA into these balls on strings. And if something is 130 million, how many balls does it take to wrap? That if you wrap 150 each time. And let's do easy math, 150,000 divided by 150. Like 100,000. It's 100,000 balls. Each ball has 8 different proteins. So that's 800,000 proteins. Each protein is, let's say, 200 amino acids. So 800,000 times 200. That's 1.6 million amino acids. Each amino acid takes 1 energy unit to make a protein. You get 30 ATP from 1 glucose. This is way more math you probably care about. These are things I think about. Why bring this up? I try to impress upon you the fact that cells are spending a lot of energy making histone proteins to wrap their DNA. And it's a pretty important process. They're spending a whole lot of energy making a whole lot of proteins just so we can make DNA take up less space. So it might seem frivolous to compact things so they take up less space, but it is a important, necessary component of the cell functioning correctly. And again, H4 can be methylated, and that is a signal for telomeres getting shorter. But when it comes to actually getting ready for cell division, we do see another change occur. The histone protein H3 will be replaced for another protein called SNPA. CNPA, SNPA. But only in a certain place in the chromosome, and it's only at centromeres. So around that centromere region, wherever it is, when the cell is ready to start cell division, or sometime during prophase or whatever, it doesn't matter when exactly, the cells are going to replace one protein with another protein. And as we're going to see later on when we talk about cell division in more detail, this SNPA protein is important in allowing the machinery to bind so you can actually move things around. So we call them kinetochore structures. So without the SNPA protein, kinetochores can't bind the DNA. Or rather, another way to think about it, by replacing H3 with SNPA, we force the kinetochores to go to a certain place on the chromosome, not wherever they want to go. We force them to go to the centromere region instead of everywhere else. The slide also introduces a very important term called epigenetic. And generally speaking, we think about epigenetic as a sequence-independent process. Most times it's a regulatory process. We're controlling something without relying on whether it's A, T, G, or C, or whatever. And histone proteins, when they bind to DNA, they don't care about the DNA sequence. They're positively charged proteins, they care that the DNA is negatively charged. That's all that's going on. That's what we call an epigenetic interaction. We don't care what the DNA sequence says. There will be examples of where DNA sequence is important. And that's, as we go forward this semester, there'll be several proteins that do care about DNA sequence, and some that don't care about DNA sequence. And right now, histone proteins are the first in a long list of proteins that we're going to talk about that don't care about DNA sequence. So we take a core nucleosome, which is a ball, we wrap DNA around it. We got a bunch of these little balls, and then we make a much more complex structure. And eventually we get to what is the fully formed chromatin, but it has to go through several different organizational layers to finally get to the end product. So you take these balls on string and we wrap them together and complex them together into something called a fiber, and then something called a loop, and then finally something called a domain. Now we're at the sort of the end product of DNA has been fully compacted in the appropriate way. And that really all it is a list. It'll be a pretty easy list. You start from a low level of organization or structure at the nucleosome, and then with the highest level or structure when we get to the domain level. And it's pretty neat looking at these models of the DNA molecule. Imagine that it actually goes together correctly. It's always sort of astounding. I don't know if you noticed, I like movies. I'll finish my quote here in a second.
So
Go ahead.
the thing that wraps around the nucleosomes, is that the double helix that's wrapping
Yes,
around?
the double-stranded helix is what wraps around. So if you want to mention another movie, I like movies. I don't know if you know. Who's seen the movie The Rock with Nicolas Cage and Sean Connery and that one guy and another guy and another guy? One of the movie, there's these nerve gas things, and these little things that kind of look like this. Watch the movie, you'll see it. Oh yeah, I know what you're talking about. Ed Harris, that's what I was thinking about. Sorry. There's the big guy that was in The Green Mile. I don't know what it is. Not Michael McDawson, the other guy. The big other guy. The car guy. What's his name? Oh well. What can you do? What is this slide about? Who cares? Right? I already mentioned what this slide is about earlier. Even though we don't have histone proteins in bacteria, bacteria still need to compact their DNA. There's just a lot more variety of how that occurs. This also is reminding you that in a prokaryotic organism, there is no membrane-bound structure. They call it the nucleus. So generally speaking, the DNA is just kind of floating around inside the cell, but it is compacted, so it takes up less space. Instead of having the typical nucleosome structure that we see in the top left, for you as you're looking at it, there are a lot of other variety of DNA binding proteins that we find in different types of bacteria that are used to shape DNA into different forms. And that's all you really need to understand and appreciate, hopefully, is that everybody needs to compact their DNA to make it fit inside whatever cellular structure or cell that you have. So this level of organization of taking the DNA and wrapping it around proteins and sticking it into a nucleus, we still have to make sure that the euchromatin parts are in the center of the nucleus and the heterochromatin parts are more on the outside periphery, that's the word, periphery, of the inside of the nucleus. So I still think it's astounding that the cell can keep track of all these things, even though it's wrapped the DNA around these proteins, and make sure that everything's in the right place. I would say at this point in the semester, we are not exactly saying goodbye to bacteria, but most of what we talk about for the rest of the semester is more of a eukaryotic specific type of content. And it starts here with talking about organisms that have different numbers of chromosomes, which means we need to start talking about the idea of diploid, because all eukaryotic organisms are diploid organisms. We need to think about what it means to be diploid and how many chromosomes does an organism actually contain. One thing that is interesting is chromosome number and size, complexity, are really not related to each other at all. Sometimes you can have the exact same amount of DNA, but have significantly more chromosomes than somebody else. You may notice from this slide here, if you can read these things, chickens have 78 chromosomes. I always joke that's why you can cut their heads off and they still move around, and they have lots of chromosomes. We don't have that many, so if you cut our head off, we're dead. And moths, I mean, it's a moth, so who cares? Step on it. But here's something fun I like. A scorpion only has 4 chromosomes, but a fly has 12 chromosomes. They both have about the same amount of DNA and the same number of genes, but for whatever reason, this one has 12 pieces of DNA, this one has 4 pieces of DNA. And humans have 46 pieces of DNA. Why does everybody have different numbers of chromosomes? Something you should think about. I can understand we all have different genes to give us different traits, but why doesn't every single organism have the exact same number of chromosomes that are all exactly the same size? I mean, if you can think of a logical reason why we need to have a multitude of different number of chromosomes, then maybe you know more about life than I do. But think about it. Why? Anybody know why these things have so many chromosomes? They're plants. Plants like to do a lot of weird things where they increase their number of chromosomes for no reason. We call it polyploidism. It's a word we'll get in a second. I'm gonna leave you with with an introduction and a tease, we're gonna talk about karyotypes just a little bit. And it'll continue on for the rest of these slides, this idea of a karyotype is really just a way that we can image or picture or view the chromosome content of a cell. How we actually get it to look like this is not important for right now. But today we will leave you with this one basic point. We can view the number of chromosomes that an organism has. And by viewing the DNA, we can spot some pretty important anomalies that we want to think about. Some of them might be chromosome number being different, or maybe the chromosomes have changed in some way. There are some really cool things, or actually really kind of bad things, we can identify based off of using this karyotype. And pick up this Friday.